He speaks he's today delivering a lecture that's titled Frontiers of Science, and this lecture has been initiated by the Indian Academy of Sciences this year, and we are inviting very prominent scientists throughout the world to come and give these kinds of talks in various parts, various institutions in India. So we are actually very happy that the Indian Institute of Science Education and Research in Kolkata has allowed us to use their premises and their hall and their infrastructure for the Indian Academy of Sciences to hold this lecture. Dr. Andy Peterson is currently the Chief Scientific Officer of MedGenome in Foster City, USA. Just a few months back, or maybe about a year ago, he was a principal scientist and the senior director of Genentech, a very famous pharmaceutical R&D company that's again located in California. He graduated, he did most of his studies at Harvard and was at Duke. His major contributions have been in the area of neurogenetics, and later he moved over to making some seminal discoveries in metabolic regulation. Most recently, he's initiated, or he's actually championed, a major study in India. He's going to talk to us about that, which has just been published. Many of us have actually participated in that study, and but clearly he's led the study from the front, and so it's a delight for us to have him deliver this lecture that's just been published on the 5th of December in Nature as a cover article. So without further ado, let me call upon Dr. Andy Peterson to deliver this lecture, and it's essentially a summary of this particular paper, and he will talk to us on benefits and challenges of diversity in data. Andy.
Thank you very much, Partha. So I'm gonna have the first part of the talk be an expanded rationale for why we've been doing the studies we've been doing, and then I'll have the middle part be about what's in the Nature paper, and then the last part will be about the studies we've done that carry forward from that are focused more on South Asia than Asia as a whole. So starting with the goals of human genetics, I'm sure we could come up with more, but so this is my list, and the list is that we want to use genetics to provide the basis for predicting disease risk, so those aspects of disease risk that are genetically determined. That's important for a number of reasons. One, for health resource planning, for a population basis, understanding what the population risk is of various diseases, but then also probably more individually impactful is predicting individual disease risk. Again, the genetic part of that risk. Many diseases, of course, many complex diseases have an environmental component as well. And then equally important are the implications that genetic associations with disease have for understanding the mechanisms of disease. We have two different means of understanding, of carrying out experiments in humans to understand disease. One is we carry out clinical trials, interventional, and the other is observational studies, and that is with human genetics, where we can look at different changed variables and look at the effects of that on disease. So in terms of the rationale, starting with the rationale for the behind the studies that were described in the Nature paper, I've got this question up here: what have genetic studies produced? There's different ways of describing this. Let's see if this. I don't know how to start the movie when I don't have the. There's a movie behind this. Oops. Oops, I didn't do it.
Should I exit and then play it?
Exit.
Playing from here?
Yeah.
Is it linked to this?
It's linked, but it should be just, there we go. Clicking on it should do it.
Probably we could try
Otherwise, we'll just
If you have the
forego it.
movie and the link somewhere.
Yeah, I'll talk over it. It's okay. I don't want to go out of the presentation to do it. Well, what the movie shows in animated fashion, and you can find it on from the link there. In animated fashion, it shows the accumulation of genetic observations, genetic associations with disease that were accumulated over about a 10-year period in the movie from about 2008, I believe, or 2007 till 2017. And in that period, the accumulated state of knowledge of genetic associations of disease went from a few tens to something like 60,000, I think, if my memory is correct. So it's a very dramatic depiction of the accumulation of knowledge that occurred over that period of time. So what was behind that accumulation of knowledge was genetic studies carried out on a large number of individuals. So this graph shows you the number of individuals included in Genome-Wide Association studies over the period of 2006 to 2018. And more than 100 million people have been studied in genetic associations of various human traits, again, primarily studies of human disease. But almost all of those individuals have been of Northern European origin. So that's shown, again, quite graphically here in terms of the different colors. And then on the other side is the proportion of the global population in those different population groups. So again, essentially 100 million out of the 125 or so million individuals that have been included in GWAS studies have been of Northern European origin. This has been pointed out in a number of different forums. I actually particularly like this one, this depiction, because most prior depictions just said European, Asian, African, and didn't subdivide Asia into any subregions. So this one at least divides Asia into East Asia and South Asia and other. But Asia is, of course, quite diverse, and there's quite a bit of subdivision of population groups within Asia that's not reflected here, quite a variation of population groups in Africa, etc. And that also highlights that, with comparison to the structure of the European population, these lower part of the graph would be divided into even more subdivisions than are depicted here. But an obvious question that comes from that is, if we've studied 100 million people, can't we just take that information and extrapolate from it to understand the genetics, the genetic basis of disease in the rest of the world's population? And I think that's a very reasonable question to ask and to try to take that apart, why that may or may not be true. So what was the scientific observations that were behind that 10-year period that's ongoing, but to be honest with you, petering out? The scientific understanding that was behind that was the recognition that individual variants in the genome tend to be co-inherited in blocks, so-called haplotype blocks, such that this is, to an approximation, a unit of inheritance, so where each colored block would indicate a variable position in the genome. And that those blocks then can be brought together in different collections. So the pinkish or the red one here could go together with the blue or the red, etc. So you can put those all together. But that individual nucleotides in the genome are not independently assorted, but rather assorted in these groups. So there was that recognition that that was a structure that was inherent in the human genome. And then the second insight that flowed from that was that by querying a subset of those variable positions of the genome, you could predict the missing variable positions. So you could impute. So this is from a review in Trends in Genetics, and then this is from a website that provides one of the major softwares for carrying this out, but essentially it's depicted here that if you sample a subset of the positions in the genome, so these are genotypes that would be produced, for example, from a SNP array, a relatively inexpensive genotyping array, that could be compared to a lookup table, a reference panel, or a reference haplotypes, and that could be then used to infer or to impute the missing genotypes. And really that was the basis for that explosion of information, using relatively inexpensive genotyping arrays that could then be used to genotype large numbers of individuals. The missing genotypes could be filled in by this imputation process. This then brings us to one of the reasons why you cannot just extrapolate from the genetic data from 100 million Northern Europeans to fill out the missing bits in the rest of the world's population. It's illustrated in two different aspects of that are shown here. An important one is the patterns of linkage disequilibrium. So in other words, the haplotype blocks that are present in the genome differ between different population groups. They differ for two different reasons. One is you can recognize a haplotype block because it has variable positions. So if some of those positions are not variable, then the haplotype blocks may appear to be different or may be functionally different. And so that's shown here with respect to the set of variants that had been associated with disease in Europeans, and then looking at whether or not those positions are variable in either East Asians or Africans. And you can see that there's a large number of variants that are variable across this allele frequency in Europeans and are associated with disease, but are essentially not variable in Africans or East Asians. So if one would guess that if the reverse was true, that we had a large number of genome-wide association studies in East Asians, you'd get a similar pattern by comparison to Europeans. But nonetheless, there are sites in the genome that between different population groups are either highly variable or are fixed, and therefore affect the apparent patterns of linkage disequilibrium. The other thing, of course, that determines these haplotype blocks is sites, hotspots for recombination. So, in other words, recombination occurs within narrow boundaries, and that then, of course, means that pattern of recombination sites is what determines this haplotype pattern, as well as frequencies. And recombination hotspots can vary between different population groups. So that's shown here with respect to different alleles of PRDM9, which is a major determinant of recombination hotspots. So common variant studies, which is what I've been talking about so far, can benefit from or require genetic studies in different population groups. One, because allele frequencies can vary significantly. In other words, there's variant sites which are not variable in some population groups, but highly variable in others. Some variants are at higher frequency in different population groups, and also the patterns of linkage disequilibrium. So in one population group, there may be a large block that's co-inherited, in another population group, a small block that can be co-inherited. So comparison between different population groups means that you can narrow down the range of genetic association and or the genetic prediction of disease risk can vary between different populations. And that's, of course, because of the population history, the number of different aspects of population history that can vary between different population groups. So one of the significant implications of this that has arisen in the last few years is because of polygenic risk scores. So polygenic risk scores have been around since the early days of genome-wide association studies, so again dating back to the mid-2000s. And the first approach that people took was just to take the genetic association signals that they recognized that met a threshold significance, and to query all of those sites. So this shows an early approach to that, where 30 different loci that affected blood lipids, HDL, LDL, or triglycerides, were queried, and an additive score was produced that did show a predictive ability for the different lipid traits. And that was done for a number of different traits. But what was recognized just in the last few years is that arbitrary p-value cutoff was limiting the power of polygenic risk scores. And so this is from a paper from Amit Kara and Sek Katarasan, where they used an approach that a number of people have also applied, where rather than limiting oneself to a relatively few loci, instead one allows all the variable sites in the genome to contribute to the polygenic risk score. So so-called genome-wide polygenic risk score. And these polygenic risk scores that are at the extremes of prediction can have very significant predictive risk. So in other words, I think it's the 1 percentile extreme of coronary artery disease risk is nearly 5 times the risk of the what you would call the general population. In other words, the rest of the distribution there. Similarly, for atrial fibrillation, inflammatory bowel disease, breast cancer, and type 2 diabetes, very strong discrimination when a polygenic risk score is produced from querying of many different loci across the genome. So I believe this one, in fact, rather than having 30 loci, has nearly 6 million loci that are contributing to the prediction of risk. Importantly, the big advance is that the fraction of genetic risk that is predictable is very significant with these genome-wide polygenic risk scores, such that in the case here again of cardiovascular risk, the genetic risk prediction from the extremes of the polygenic risk score are equivalent to the risk that's produced by very high impact loss of function variants. So in the case of LDL cholesterol, loss of function for the LDL receptor causes familial hypercholesterolemia and very significant risk. Similar increase in risk is produced by the extremes of polygenic risk score. So very strong, in ideal circumstances, very strong discrimination between different genetic risks for disease. So this then gets to the heart of the question of can we not just extrapolate from the genetic studies that have been done in Northern Europeans? Can't we just use those studies to predict risk in other populations? And that has been tested very directly. So the two things that one needs to do are to, at face value, to at least correct for allele frequencies, and then also to correct for patterns of linkage disequilibrium. So in other words, you may have 6 million SNPs, but you don't want to be double counting haplotype blocks. And also you want to be looking at sites that are actually variable in a population at the appropriate frequencies. So if one does that, so if you carry out generate a polygenic risk score and compare it or assess its predictive ability, if the data, the underlying data comes from Europeans, which is, as I said, given the bias is the only state that we have, starting state, and then you use that to see how well does it predict actual risk in different population groups, you can see that the ability to predict risk falls off very dramatically, even though the allele frequency and the LD patterns have been appropriately compensated for. That then tells you that the actual impact of all the individual variants, the 6 million variants that make up that, vary between different population groups. And the only way to account for that is to carry out a GWAS study in the appropriate population to produce the appropriate polygenic risk score. So that then is a clear and graphic demonstration of why we cannot extrapolate from the studies in Northern Europeans and apply those findings to other population groups. I want to point out that there is a kind of both a functional and a conceptual dichotomy in different types of human genetic studies. A simple one is between common variant studies, which is what I've been talking about so far, and rare variant studies, which, depending on how they're structured, can be carried out differently, but also can give you different types of information. So there are also quite clear reasons why it's not possible to simply use studies from Northern Europeans and apply them to other population groups. So the thumbnail sketch of the population genetic or population history theory behind rare variants that's critical for the discussion today is that the rare variant, the site frequency spectrum for rare variants has been produced, or the pattern that we see has been produced by a rapid population expansion. So what we understand from the expansion of the human population over the past centuries and thousands of years is that following the development of agriculture, when the shift from hunter-gatherer lifestyles towards agriculture occurred, this allowed a rapid expansion of the human population. And what that then allowed to happen is the accumulation of rare variants. So in other words, rare variants occurring that might reach equilibrium over a long period of time, instead have not had the chance to reach equilibrium. Common variants apparently have existed for a longer period of time and have reached equilibrium. So why is that important? It's important because the various population groups, major population groups across the globe, were resident in their geographic sites at the time of which these population expansion occurred. So in other words, the pattern of rare variants that one sees in a population is unique to their history. It occurred after they had migrated from the populations had spread throughout the globe. After they had spread throughout the globe in a hunter-gatherer lifestyle, then agriculture was introduced. The populations expanded rapidly, and the accumulation of rare variants occurred, or the spectrum of rare variants occurred. So rare variants are more often, or I should say, are enriched in functionally important rare variants. So in other words, protein-altering variants. And so this comes from one of the earliest large-scale exome sequencing studies, where they described variation between African Americans and European origin Americans. And they described that 95% of the missense or loss of function variants were, quote, rare, which I think in their definition at the time was less than 5%. That definition varies somewhat. And that 82% of those rare variants were found only in one of their population groups and not in the others. So in terms of the general picture, that the biological impact, the likelihood of dramatically altering protein function is higher for rare variants, and for common alleles that have been subject to a long period of purifying selection, the effects are typically, the biological effects are typically much lower in magnitude. So obviously, in terms of understanding the biology of any particular human trait, the rarer variants are very important and very important for our understanding of the genetic basis of disease. So again, if one only studies Northern Europeans, one is sampling a narrow set of variation in the human genome, and one is then not capturing the alleles that are contributing to disease. The rare variants that contribute to disease in Europeans are not the same as the pattern of rare variants that contribute to disease in other population groups, South Asia, Africans, or other groups. Also important, and then we'll come to later, is that the likelihood of homozygosity of rare alleles varies quite dramatically between different population groups. So coming back to those goals for human genetics, predicting disease risk and then understanding the biological basis of disease, bringing more population groups into genetic studies is very important in terms of being able to predict the basis of disease in those other population groups, non-population groups. And that then, of course, there are, to some degree, gene-environment interactions. And again, the environments are going to be different, et cetera. So that's something that I haven't brought up, but is related to the genetic prediction, so that improvements in healthcare delivery are made in an appropriate fashion based on the genetic risk, the environmental risk, et cetera. And finally, regardless of the specific population group, the understanding of disease and the ability of genetics to help illuminate the basis of disease is missing out on insights by only studying one pool of rare variants in one population group. So I just want to touch on this last point here, that there are also important differences in population structure, and this will come up towards the latter part of my talk, that differences in population structure can affect the structure of the genome, or the structure of the genome of a population. And there are attributes of that can provide powerful opportunities for discovery. So that can differ between different population groups, in addition to the rate of rare variation and the pattern of rare variation. So that's illustrated here in terms of characterization of Finland. So Finland and Iceland are well recognized as founder populations. So Iceland being the one that's most intuitive. A few individuals founded the present-day population of Finland, such that if a variant could have been present at 1 in 1,000 individuals in the starting population in Scandinavia, if one of the 10 individuals who started the population in Iceland carried that, it would then become a 10% frequency. So that's a bit of an exaggeration, but that's the basic prediction. And so you see that to be the case in the Finnish population. So it's shown most graphically here, where the frequency of rare variants at this end of the graph here are decreased in Finland's as opposed to non-Finnish Europeans, and that there's a corresponding increase in variation at higher frequencies. And this is shown specifically for protein truncating variants, missense variations versus and synonymous variants. So you see this pattern of things that are low frequency are depressed, of course, because if you're starting out with only 10 people and expanding that into the large population. But functionally, that's important. It's important for carrying out genetic association studies because, as I said, these what we're calling rare variants, protein altering variants, are enriched in functional variants. If they're present at 1 in 1,000, then to power an association study, you need a large number of individuals. If they're present now, if that frequency is shifted of that missense or loss of function variant is shifted to 10%, then one needs a much, much smaller study size to detect the effects. And so that's shown here with this graph here, showing that these are variants that are discovered to have an effect on a number of different traits in Finland. And you can see that there's an enrichment for alleles that are present at a higher frequency in Finns versus non-Finnish Europeans. And so this is a phenomenon that's been capitalized on in Iceland, in Finland, and in various other population groups to aid in the understanding of disease. So, and that's the article that figure came from. So it's worth thinking about then, well, why does this situation exist? Because if we want to change the situation, of course, it's useful to understand why it exists. I'm sure we can come up with more factors or more reasons why this bias in the inclusion in genetic association studies. I'm sure we can come up with more, but this is at least a high-level set. One is obviously resource limitations, and that comes at many different levels. The available money, the available public funds that are devoted to science in different countries in different parts of the world, the available resources, the history of infrastructure and training, and et cetera. There's many different factors there in terms of research limitations. There is personal privacy, an important concept that quite often means that to control the privacy rights of individuals, including genetic privacy, data use is limited to within or is controlled within national borders, and so that limits international collaboration. So the type of studies we're talking about often include large international groups of scientists working together. So in as much as that these privacy rights limit data sharing, et cetera, that's a reason. And finally, what I'm calling network effects or feed-forward effects. In other words, the more characterization there are of Northern European genomes, the easier it is to carry out, and the more powerful the studies are in Northern Europeans. It just gets increasingly easier. So that then increases the problem. So thinking about this, what are the things that we can do something about? So what I came to in thinking about this is technically the most tractable part of all of this to try to make a difference. So that led to the involvement in Genome Asia Consortium. So as Partha indicated, this was published recently in Nature. It's a project, it's a loose consortium, it doesn't have its own funding, it's self-funded by the individual members of the consortium, and really bound together by the idea of trying to produce approaches and tools, data sets that will make it easier to do genetic studies in other populations. So in other words, rather than just coasting along and letting the available data sets determine where we do additional studies, let's generate tools, et cetera, that will facilitate doing genetics in other populations. So there is, of course, a project that was done some years ago, 1,000 Genomes, that was designed to increase our understanding of haplotype structure in the genome from various parts of the world. It was fairly low resolution, 1,000 genomes from all across the world. It grew to about 2,000 genomes. But what we set out to do is, in this pilot project for this Genome Asia, was to carry out essentially what I'm calling loosely 1,000 genomes for Asia. So focusing in on primarily just on Asia. So you can see the distribution of the number of individuals from various parts of Asia that were sampled. We did not try to sample deeply from everywhere. There were practical limitations in terms of where we could obtain samples, and where also there were existing data that would be available, et cetera. And at the same time, we also sampled from other parts of the world. So this is important for internal comparisons, for comparisons to external data sets, for studies to understand population history from within the data set. It's not sufficient to study a narrow part of the world, but rather to sample intensively in Asia and have lower level sampling from other parts of the world for comparative purposes. So there's a number of different implications for this. One is, unlike a number of projects that have been done to produce reference sets like this, The samples that were sequenced here were from geographically defined origins. So in other words, these were not expatriate populations. These were not individuals that, had, for example, had migrated to a different part of the world before they were sampled. And what that allowed us to do is to look at allele frequencies with respect to the geography of the world. So in other words, the populations that are resident in those parts of the world today. So this shows the geographical distribution with respect to the distribution of predicted adverse responses to carbamazepine. So this is an anti-seizure drug. There are known drug metabolizing enzymes that affect the likelihood that you will have an adverse response. And you can see that the predicted frequency of adverse responses to the standard dosage varies dramatically, with a high level of adverse responses predicted in island Southeast Asia here in Indonesia, and essentially no predicted adverse events in Northern Asia. So it can vary quite dramatically. And again, this is important. So for example, warfarin in the US has an indication that the dosage should be adjusted for Asians with no kind of discrimination in terms of what that means. So in other words, does that mean Southeast Asians? Does it mean East Asians? Does it mean South Asians? And it's not indicated. So obviously the actual most useful is to have individualized pharmacogenomic information for the individual before prescribing. That's not always possible to do, and so having geographic predictions is useful. In the project we had, thanks to Partha, the sample collection that he had put together at NIBMG over many years, we had a high density of sampling in South Asia, such that we had an even finer description of the population groups as well as the geographic location, and that's shown here on layered on top of information about the language family, which is spoken by the groups that were sampled, as well as the type of population group that was sampled. So that again allows us to have to be able to map the information that we had. So for instance, there's an allele of beta globin that is associated with beta thalassemia. We picked out this allele because it is essentially non-existent in most of the rest of the world. So you can see here, at least within our sampling set, the only place that the allele is, the only appreciable allele frequencies are South Asia and Southeast Asia. And in South Asia, of course, we can see that it's distributed at higher frequencies within some population groups than others. So there's a number of types of analyses like that can be done. That data is useful as it accumulates in larger and larger size. But coming back to the point of how do we build tools and approaches that will allow us to shift the advantage of doing studies in Northern Europeans. We also put together a reference panel for imputation. Excuse me. So it's present at the University of Michigan imputation server, which is free for anyone to use. You have to register, but then after that you can use this Genome-Asia pilot project imputation panel. So a collaborator, Hilary Martin, at the Wellcome Trust Sanger Center, carried out studies comparing the effectiveness of imputation with this reference panel versus what has been considered the most diverse and most useful general imputation panel, the Haplotype Reference Consortium panel. So Hilary and Chinchin compared imputation with the HRC reference panel with the Genome-Asia reference panel, and then used as a truth set exome sequence data from individuals of Pakistani origin that were living in the UK. That was for convenience of what they had. And then they used, those same individuals had been genotyped with a SNP genotyping array, and then they imputed using either the HRC or the GASP. So what's shown here is, and what the measure that they used throughout all this is what they called non-reference discordance, if I'm remembering it. So it's mismatch between the exome sequence genotypes and the imputed genotypes. So the rate at which the sites are discordant per sample is lower for the Genome-Asia panel than for the HRC panel. That's shown here with respect to a scatter plot where it's shifted, the axes are not the same, but it's shifted to the left and up, as you can see. And then they also looked at binning from different allele frequencies, and you can see that the per SNP discordance rate is higher, again, for the red bars, the HRC imputation, than for the GASP imputation. What's actually quite a bit more dramatic is that quite a few more variable sites were imputed by the HRC reference panel than by the GASP panel. So they asked the question of the sites of the variants that were not imputed by GASP and were only imputed by HRC, what was the rate of discordance? So that's compared here where it says imputed by GASP or not, and what that means is imputed by both or not imputed by the GASP panel. And you can see that the variants that were imputed only by the HRC reference panel were actually quite likely to be wrong. So in other words, the increased number of variants that were being imputed were enriched in poor imputation. This is not where we would expect to see the greatest benefit from this reference panel, because this is, of course, individuals from the northwest part of South Asia where have a higher, closer relationship to Europeans than South Indians do, but nonetheless the improvement is significant. So the question then is, how do we further lower the barriers for human genetic studies, with the question of how do we do that specifically in South Asia? And I want to bring up, in addition to the points that I already brought up, a couple of ones. One is that the statistical methods that we are commonly used have really been based on the population structure of Northern Europe, because that's where most studies have been carried out. So the population structure there is generally modeled as a large, recently expanded, outbred population. So is that true of all the world's populations or not? So what is the population structure of South Asia? This is not a question that I can answer. I'll leave this to people like Partha and Annalaba to answer, but do we need to optimize methods so that they are more optimized for other population structures, specifically for South Asian population structure? And obviously we need to understand the population structure to answer that question. And then finally, coming back to the same point that I raised earlier, can we build even more tools that will facilitate genetic studies in South Asians? So looking at this first question, we did looking back then at what's already known, again from studies by a number of people, including Partha and Annalaba and Thankaraj at CCMB and other workers. We have a picture, and this comes from a model of the origins of South Asian populations from David Reich's lab. We have a picture of the South Asian population structure of ancestral North Indians, ancestral South Indians, some component of Austroasiatic, and then minor components of Tibeto-Burman populations, etc., that came together and formed individual population groups that coalesced about, let's say, 5,000 years ago. That picture has been a relatively consistent one. Of course, the details vary between different studies, but has been a reasonably consistent picture, but has been based on essentially SNP array studies from well-defined isolated population groups, a large number of different population groups, but nonetheless, studies have been chosen on the basis of representing population isolates. And that picture then, has a number of implications for genetic studies. One is that South Asian origins are quite different from Europeans or East Asians, and so that there's going to be genetic variants, rare variants, as I said, as I described before, that are found only in South Asia. There's painted the picture of thousands of different population groups, each of which captures a set of genetic variants, that those individual population groups quite often have strong founder effects, not because of geographic isolation, but because of reproductive choice within the group, and that there are patterns of endogamy and in some population groups consanguinity that increase the rate of homozygosity. So again, I want to emphasize that this picture has been consistent, but has really been focused on studies in well-defined isolated population groups. And in the paper that was published in Nature had that same selection bias. So in other words, the population groups that were studied from South Asia were primarily from defined isolated population groups. We did, however, include a couple of samples, one from Chennai and one from Bangalore, right over here, that were not from any particular population group. They were just people who came into a hospital. And so what we've done here is identify, use a measure of founder effect, identity by descent. We used the Finnish population, which is a well-recognized founder population in Northern Europe. We used that as our reference point for strength of founder effects. And so you can see that there are a large number of groups that have less of a founder effect, so more outbred population than the Finns. There are a number of groups that have much stronger founder effects, and these tend to be groups that have very, very small population size, so it's not surprising in hindsight that they have such strong founder effects. But metropolitan Chennai has a population of 9 million, and this sample was essentially a random sample of people living in Chennai. So that was somewhat surprising and begged the question, which is, does the general population of South Asia, and importantly for carrying out disease association studies, how does the general population of South Asia relate to this population, to this picture that we have developed over time from study of well-defined population groups? So how different or similar is it to the picture that we got from prior studies? And that's important because it has clear implications for how you think about carrying out disease association studies. You need to carry out disease association studies by studying people who come into the healthcare delivery system, and if you need to subdivide, put labels on people as being from one particular group or another, then that's very different than if you can ascertain them based on disease. So this slide paints out the picture, the general picture of the samples that we used to characterize this. So what's shown here as kind of a reference framework for genetic diversity is the principal component analysis of the samples, the non-South Asian samples in that Nature paper. And so you can see that this spreads out the diversity between Europe, East Asia, Africa, Oceania. This kind of defines the overall global coordinates for genetic diversity. If we then take the South Asian samples from that study So the about 600 people that were from various South Asian population groups, and we layer that on top of this, shown in red, you can see that it covers a big swath of the genetic diversity that's defined by these global axes. And then if we look at 5,000 people who were ascertained solely because they came into the healthcare delivery system from the northwest, the northeast, and the south of India, you can see that they cover a large chunk of genetic diversity as well. And you can see also population substructure within the various groups. So in other words, from West Bengal or Bangladesh, there's substructure. And if you look at Chennai, I pull it out, it's kind of lost in the back behind these other ones. You can see that there is population substructure in Chennai. There's at least 3 apparent population groups that are present in that healthcare delivery, in that diabetes hospital in this case, where these were sampled. And I would point out, in case it's not apparent, the separation that one can see does not imply some degree of reproductive isolation to have the genetic drift that would produce these differences. So focusing in a little bit for the next slide or two on samples from West Bengal, from a rural area, Birbhum, north of Kolkata, we just focusing in on those samples from that study area, one can see that there is significant population structure. It's spread out here, the PCA analysis carried out just on these samples. There are at least 1, 2, 3, 4, and probably 5 different population groups that one can identify in this area. The question is then that's relevant to thinking about all the types of studies, rare variant studies, and founder effects, etc. One question is, how long has this population structure existed? There was an analysis carried out by looking at private rare variants. So in other words, the rate at which rare variants were occurring in each individual population group. And then also looking at Bangladeshi samples. And then predicting putting that on a time frame in terms of how long it's been since the groups diverged. And so each of the comparisons is to the general caste group in West Bengal, and you can see that the Bangladeshis have been separated, reproductively isolated for about 700 years or so, and then the various different groups have been separated from each other for times varying from maybe 100 years or so up to 3,000 years. So quite long separation times. That again calculated from the private rare variants. And then another aspect that we characterized was the rate of homozygosity in the genome. So this has been observed from SNP variant studies again, but has not been characterized in large sets of individuals from the general healthcare delivery system. So it's been well recognized as an attribute of population structure in Pakistani populations. So I've got the samples from Pakistan as a reference, and then that's compared to individuals from Chennai and Bangalore in terms of the rate of excess homozygosity. So in other words, how often is a particular site homozygous compared to what you would predict if there was a completely outbred population. So the Han Chinese are shown here, smaller sample set, but they deviate a little bit from a totally outbred population. But the rate of these samples at low allele frequencies, these individuals, is 50 or 60 times that predicted from an outbred population. Because we were looking at whole genome sequence, we could also carry out an analysis to look at what the parentage is that leads to this rate of homozygosity. And we know about the patterns of endogamy, marriage within the group, or consanguinity in some population groups. And we could then divide those into both of those processes using the second cousin parentage as a surrogate for endogamy. There's no biological definition of endogamy. And then also first cousin equivalent as a definition of consanguinity. So the population structure, I think that's very clearly indicated as being different here from this kind of picture. So one question is, do we need to be using different methods for carrying out effective genetic studies in South Asian populations? And so I'm just going to end with the second generation of tools that we've developed. One is, so that reference panel that I showed you first was based on 1,700 genomes, added to substantially from adding to it from South Asian specifically additional samples, such that it's a much larger reference panel and very enriched in South Asian genomes. And then subsequently developed a genotyping array that is optimized for South Asians. So a commonly used genotyping array, the Illumina GSA array, we've looked at the sequence data, and 90% of the functional variants that are present in South Asian genomes at 0.1% or higher are not capturable by the GSA chip. So with that in mind, working with Thermo Fisher to design a genotyping array that is optimized both in terms of the imputation backbone, in other words, the backbone that allows you to carry out effective imputation, is optimized for South Asians, and then directly genotyping functional variants down to 0.1% as best we can. That's our kind of our stretch goal, is to hit that, and then have that direct genotyping at low frequencies, again, working and optimized with the imputation backbone. So it has a number of different potential uses, but importantly, it should be a much better tool for carrying out large-scale genome-wide association studies than what has been available so far. Importantly, I do not want to forget to acknowledge all the people who contributed to all this. Jeff Wall played a huge role in all the analysis, together with Eric Stowiski and Ravi Gupta, and more recently, Fas Satirapong Sasudi. And then important contributions in terms of the sample sets that I've talked about throughout came from Partha Abhijit Chaudhuri, Anamitra Basik, and Rajesh Kumar in Birbhum for the Birbhum study. And then all the members, there's a very large number of individuals who contributed time and effort and analysis as well as samples to the overall consortium. So I will stop there and I'll be happy to take questions.
Good evening, sir. Santi from Sastra University. Is there any temporal data set is available attached with this genome for predicting that risk, disease risk?
Yeah, thank you. Excellent question. So the data set that's described in the Nature paper, as I said, was primarily from isolated population groups. So those were collected, ascertained based on who they were, and they we didn't have phenotypic clinical trait data associated with those samples. The subsequent samples, yes, most of those we do have, for instance, have blood chemistry and other measures associated with them. So that's analysis that can be done. Does that answer your question?
Hi, Andy. So the slide that showed that when you try to look at the genome-wide polygenic risk score, based on the data from the Northern Europeans,
yeah.
it did not at all predict to any level in other populations.
Yeah.
So does it surprise you, not because of the technical reasons, but the biology, the mechanism, the biological mechanisms of the disease, would it be dependent on different populations, and most populations live in different geographies, and therefore an environment interaction? Does it discourage us to think that, you know, the GWASs that could be done in South Asia could have more problems because of that, because we do not have a homogeneous kind of background?
Well, so one point I want to make is that the risk is a population risk, so it's a relative risk within a population. So even if a risk is higher within one population, even if, say, the risk of diabetes is higher in South Asians than in Europeans, that's not the comparison, because you're comparing relative risk within the population. So I don't know if that was part of your question.
No, so what I mean is, so if you take the common variant, common disease hypothesis, so the common variants are supposed to be, in a particular disease, you'd have some major genes, some minor genes, and wouldn't that be conserved for a given disease anywhere, irrespective of the population?
No. This is a short answer. There's a large degree, there is overlap, but the overlap is not sufficient. So there's there's some difference in allele frequencies. So if you have an allele that's associated with disease risk, say, but it's at much higher frequency in a population, you might argue that that shifts the whole population. But like I said, we're comparing relative risk within a population. So then also the linkage disequilibrium patterns vary. But then importantly, the real point is that the effect size of these variants is different in different populations.
Different populations, yeah.
Yeah, and that's why that after you correct for allele frequencies and linkage disequilibrium, the effect size is the only way to correct for that is to carry out a study within the population.
But you have included SNPs genome-wide, right? So that would take care of meaning, wouldn't that, because you're covering genome-wide, so you're including every SNP that would play a role in the pathway of a disease?
You only want to count each haplotype block once, and you want to count it with the variant that has the largest effect size. So so in other words, if that varies between populations, you're gonna lose predictive risk.
So if there are no other questions, you have a question on? Somebody else has a question? Yeah, sure.
Thanks for the talk. How do you select the individuals from the group of populations? I'm just curious.
How to select them from?
Individuals. How do you select the individuals from the population group? Means for sampling?
So
for data collection.
For the last part of it, the talk, because it's different from.
Yeah, the last part of the
Yeah. So for the last part of the talk, the individuals were selected on the basis of disease. The individuals from Pakistan were selected for a case control for cardiovascular risk. So some of them are controls. In other words, they did not have cardiovascular disease, and other ones did have cardiovascular disease. In the samples from Bangalore and Chennai, some of them have diabetes. Some of them have eye disease. The individuals from West Bengal are from an epidemiologic study, so it's a community-based study, so they're, in the general population. But the point is, so there absolutely can be some sort of ascertainment bias in that collection. But the point is that they were ascertained purely on the basis of they were in the healthcare delivery system. There was no kind of, who are you, what is your, what is your last name, what language do you speak, nothing along those lines. It was just, so in other words, it was representative of individuals that you would recruit into disease association studies.
Okay. Thank you very much, Andy. That was a fantastic talk, and I suppose that each one of us has enjoyed the talk and learned quite a bit. May I request one of the senior professors of this institution, to present a bouquet to you, Professor Narayan Banerjee.
Thank you. Thank you. Wow. Thank you very much. Thank you.
Thank you all for coming to this talk.